the idea of transplanting organs from animals to humans goes back at least to the 1960s, where a chimpanzee kidney survived for 9 months in a school teacher who went back to teach, and was normal for 9 months. But that was the exception then, and it would be the exception now, except for the synthetic biology that we do on the germline of pigs, which now are made it into many preclinical primate transplant trials, pig to primate, and a few pig to human trials that are going on. Primate survival looks like around 600 days so far, and they're still a couple of them are still alive at 500 and 600 days. But we're keep improving these. But that's in the order of 40 to 60 edits per genome in the germline. If the multivirus resistance requires more than that, some things that are done for diversity and ecosystem maintenance may involve even more. There are a type of tape recorder, sometimes called a flight recorder, so it's analogous to planes that record a lot of data, but typically you don't read it. So a lot of writing, not much reading, unless the plane goes down, and then you'll look at selective regions for debugging what went wrong. That same thing could be put into the bodies of plants, animals, and even humans, because it's a very compact recording device of the physiological states of every cell in the body. And we've shown this works sort of in the scale of 60 to 24,000, and that's probably our first effort at making 1 million edits will be in the form of these molecular flight recorders. So those are a few examples, but the number will grow as soon as we get more than a handful of people working on these visionary projects. What we'll see a blossoming of all sorts of creative uses of making multiplex editing. I think non-multiplex editing will become the exception.
So as you mentioned, your lab, gene edited pigs, and you enhance them by making them resistant to some retroviruses. Do you think, as a more visionary kind of question, that you could use, more precise gene editing, the deaminase or CRISPR, whatever, to eliminate viral spillover events from livestock to humans? So, there's a lot of viruses that originate from livestock when we're raising animals in captivity. So,
yes, this is important. So the viruses that we got rid of were endogenous retroviruses, meaning they're built into the pig genome of every pig on the planet, and they have been shown to infect human cells and to replicate and go into other human cells. So this is particularly bad scenario in immune compromised patients, and the FDA recognized this decades ago, and really was, I think, pleased to see progress being made on eliminating them from the germline of the pigs. So that's that there. But in addition to viruses that are built into the germline of animals and humans, there are viruses coming in from outside. And we just published the first example. This is with Luhan Yang's team. She was a graduate student and a postdoctoral fellow in my lab, and co-founded eGenesis and Kihan for making cell therapies and organ therapies. But anyway, as a side project, we published a paper on getting rid of African swine fever virus by making CRISPR to attack the viral DNA. This is what CRISPR originally evolved to do, is to take out bacterial viruses. We think this is the first case of using CRISPR in a practical sense for eliminating mammalian viruses from the environment. It's using CRISPR against mammalian viruses. So But zoonotic diseases is bigger than that. If we could make a huge fraction of plants, animals, and humans resistant to those viruses because of their genetic code, that actually anticipates viruses we haven't even seen yet. It should handle all natural viruses. So, like Marburg, Ebola, HIV, CRISPR. These would not have been surprises. They would have been surprises to scientists, but not to these virus-resistant cells.
So it sounds like CRISPR seems to be uniquely positioned for that type of use or clinical
Well,
application.
not necessarily. So I don't. I love CRISPR. I personally benefited from it, but it is. I like to balance it. There are other nucleases that some people claim are more specific, less off target. There are deaminases that don't involve CRISPR. So I wouldn't say term unique is too strong. We have a lot of tools in the toolbox, and a lot of it has to do with delivery and testing too. Testing is a big deal, which is somewhat swept under the rug when it's just like all we have to do is design a CRISPR and take care of everything. But there's a lot of reading and synthesis, which isn't CRISPR, and then the delivery and testing. So it's a integrated whole that doesn't require CRISPR.